Hello everyone, my name is Hubert and welcome to Teach Me channel. Today we will be learning about GCSE biology, binary fission. Now, what is binary fission? Binary fission is a process by which bacteria divide, by which prokaryotes in general divide. Last time we were looking at mitosis, which is a way eukaryotes can divide by dividing. And binary fission is the way prokaryotes can do it. And if you watched the mitosis video, a lot of the things you're gonna be seeing today is gonna be very familiar to you. So if you haven't watched it, you might wanna watch the video. I put it down in a link in the description below, so check it out. Okay, without further ado, let's get started. Okay, so what happens in binary fission? Well, first of all, we have our bacterium here, our prokaryote. So we have our prokaryote here, and it has the circular DNA, and it has some plasmids. Okay. So that's our little bacteria. So what has to happen first? First of all, as with mitosis, well, if we are going to be splitting this guy in two, then we need to replicate the DNA. We need to replicate everything here. So first of all, we need to replicate the genetic material. So we still got the circular DNA here, like so. And we still have the plasmids, like so. But at the opposite end of the cell, we have a replicated version of that genetic material. And those plasmids also get replicated. So that's what happens, and also notice after the replication, the genetic material moves to the opposite ends of the bacterium, the polar ends of the bacterium. I'm going to write it down here. So this is important because, as you can probably guess, we are going to be splitting it along this line. So that's why we need the genetic material at the polar ends. So it is very easy to split it down the middle. So we have nothing in the middle. We have everything at the ends of that prokaryote, of that bacterium. Okay. So what's next is we get a division of cytoplasm. So we have cytoplasm sort of starting to form these sort of creases here, and we have the genetic material still contained on the opposite ends, like so. Okay. Okay, so what's next is simple. Very similar to mitosis. The bacterium splits into two independent, separate daughter bacteria. Like so. So one of them has the replicated stuff, and one of them has original genetic material. Now, the very important thing to notice here is that these two cells will be identical for the time being. For the time being, they are identical. Later on, they may not be identical anymore, if they start sharing their plasmids with other bacteria. However, as soon as they divide, they are identical, because the replication of genetic material means that the genetic material on both sides is identical. So this is very important. They are identical. Okay, now why is this whole process? So this is the whole process. It's very, very simple. The prokaryote don't disappoint in this manner either. Prokaryotes are simpler than eukaryotes, and this process essentially is simpler than mitosis, because we don't have to deal with the whole nucleus disintegrating and then reforming, because everything is just freely floating in the prokaryote. So we don't have to deal with any of the sort of nuclear shenanigans. Okay, so this is much simpler. But why is this process called binary fission? Well, this is because this is actually a good way to describe what's happening here. So we have binary, which sort of means two. It doesn't mean exactly two, but it kind of means two, that we have two options. Binary tends to mean that we have two options. For example, in computing, binary language consists of 0 or 1, because we write down numbers using only 0 and 1, which essentially are two options. You can either have 0 or you can have 1. Same here. Binary just gonna mean 2. And why is that? Because we are splitting that bacterium in two. That's literally why it's called binary, because we are splitting that prokaryote in two. And fission, if you do physics, you might already have come across this word. Fission means splitting. For example, nuclear fission in physics means splitting an atom. However, here, fission means splitting a prokaryotic cell. So fission means splitting. So binary fission, long story short, binary fission essentially means splitting in two. A process of splitting a prokaryotic cell in two. Okay, so this is it on a process. However, there is one more thing that we need to cover. So we need to cover some calculations. What if... Now, the very interesting thing about prokaryotes is that because of the simplicity of this process of binary fission, some prokaryotes can divide as quickly as they can do a split every 20 minutes, which is insanely quick. So they can split every 20 minutes. So that doesn't mean that this is the case always, because this is very dependent on the specific microorganism, and the conditions need to be favorable for this to happen. But, you know, splits of every 30 minutes are actually pretty common, and this is still impressive. This is still incredibly quick compared to normal human cells, which is how bacteria can just multiply their numbers insanely quickly. They can do that so quickly, and we need to know how to calculate this. So what if we have one bacterium which divides every 30 minutes, let's say, and it divides for 2 hours. So what happens if that bacterium does it? What happens if one bacterium divides every 30 minutes and it does so for 2 hours? How many bacteria do we get at the end? How do we calculate this? Well, first of all, we need to calculate how many minutes in 2 hours. So, first of all, how many minutes in 2 hours? We do this by doing 2 hours times 60 minutes, because we have 60 minutes in an hour. So that is 120 minutes in 2 hours. Then we need to figure out how many divisions that bacterium will undergo in that time. Now we know that bacterium divides every 30 minutes. Number 2, we need to figure out how many divisions. And we do this by 120 minutes divided by 30 minutes, because every 30 minutes we have one bacterial division. So 120 divided by 30 minute units, and that will give us four bacterial divisions. And now let's figure this out. So what happens first is we have a bacterium and it splits into 2, so that's first division, and then each one of those splits into 2, that's the second division. Each one of those splits into 2, so that's the third division, and each one of those splits into 2, which is the fourth division. This is what happens, but as you can see, it's a huge mess to work this out. So we could do this, you know, if you had time and you wanted to count all of these up, you could do it this way. But much better way of doing this is essentially, what we're having here is 2, which is multiplied by 2, and then the result of that is multiplied by 2, and the result of that is multiplied by 2, and the result of that multiplied by 2. So essentially what we have here is 2 times 2 times 2, 2 times 2 times 2 times 2 times 2, which is 2 to the power of 4. 2 to the power of 4. So we just need to work out what that is. And 2 to the power of 4 is 16. So after 2 hours, we go from 1 bacterium to 16 bacteria in just 2 hours. So that's why they can that's why you can see how they can divide so quickly. Now if we added 1 more hour to that, we would have another 2 divisions happening. So this is insanely quick. So this is it for today, guys. Thank you very much for watching my video. I really appreciate it every time you guys watch these videos. If you want to stay up to date on daily science, maths and biomedical science videos, hit the subscribe button below. If you like the video, give me a big thumbs up. And if you have any questions at all, shoot me a question in comments. I will answer as quickly as possible. I love answering your questions. Thank you again, and see you next time.